Please.
I
welcome and thankful. Please, the participants, please mute your mic. Today the talk will be about 30 to 40 minutes. Then we have question answer session. The topic is very interesting for the students of zoology, veterinary science, wildlife, and other different life sciences students. So I will request the participants, please listen carefully and write down your question. If you can write down in the chat room, we can also take that questions in the question and answer session. All this talk will be recorded and will be uploaded on our DS channel. You can, if someone miss, your colleagues can miss, that can also take benefit from that recording. Now I will not take more time. I will give the mic to the speaker, Dr. Zafarman. Please continue your talk and please check either you can access to screen or not. And I will request the admin data center, please allow the speaker to share the screen. That's all, please continue your talk.
Thank you, sir. Thank you very much, sir. Thank you, sir, for your time and introduction and arranging such a wonderful series of the lectures regarding the different aspects in the agriculture and the veterinary sciences and in animal sciences. Thank you so much. My today's talk is Newcastle disease virus, or you can say Newcastle disease in poultry, its current scenario, and potential strategies to control it. I have used my affiliation of PhD because most of the work presented here is from my PhD work or from our team. That is Shanghai Veterinary Research Institute, Chinese Academy of Agricultural Sciences. In today's discussion, briefly, I will describe about the background, then current scenario of the Newcastle disease in the world, in the Asia, in the American subcontinent, and in Africa. Then I will spend most of my time that how Newcastle disease virus interact with the innate immune system and how it can escape from the innate immune responses. In the third part, I will describe about the adaptive immune responses induced by the Newcastle disease virus. Then I will describe the future pandemics that may be possible in the near future, maybe in the next 10 years or the 15 years, then how we can control it, and then at the conclusion. Everyone, I think most of the veterinary students knows that Newcastle disease caused digestive, respiratory, and nervous symptoms or the nervous disorders, and it can cause 100% mortality in specific pathogen-free chickens. But in vaccinated chickens, mortality varies depending on the antibody titer, biosecurity, bird's health status, then breeds and genetics. A lot of work have been done on the genetics or the genetic resistance against the Newcastle disease in the current year, especially in the last 4 or 5 years, and all the work done is by the scientists from the UC Davis and Iowa State. But in case of outbreak in the commercial flocks or in the vaccinated flocks, NDV or Newcastle disease leads to stunted growth and decreased egg production and cause mortality that may vary from the 0% to 30%. Even I know some flocks in Pakistan where the mortality may be 0, but after 4 or 5 days of the outbreaks, they came to know about the nervous science, and they then came to know that the decrease in egg production, especially in the breeding flocks, where the excessive vaccination schedule, the excessive vaccination is practiced, mortality is very less, but it can cause the decrease in egg production, decrease in fertility. When we look at the Newcastle disease virus, it belongs to the genome. It is caused by all the Newcastle disease, or you can say the Newcastle disease outbreaks in the whole world are caused by the avian Ebola virus serotype 1. And there are 4 different categories of the Newcastle disease virus on the basis of the pathogenicity, and those are velogenic, mesogenic, lantogenic, and asymptomatic enteric. I will not go into the detail of these categories because most of the listeners, I think, know these categories. Here is a brief history of the Newcastle disease. Almost 4 pandemics has been caused by the Newcastle disease virus in the whole world, but the current one is due to genotype 7, and that was started from the ducks in China. Newcastle disease virus is a single stranded, negative sense, non-segmented, and enveloped RNA virus, and the genome size is 15.2 kb. It has a 6 protein and 2 non-structural proteins, 1 serotype and 2 classes, class 1 and class 2. And from these proteins, the most important ones are F and HN because these are mainly involved in the receptor binding and the pathogenicity. Until now, almost all the outbreaks or all the pandemics are caused by the class 2 viruses of the avian Ebola virus or the NDVs. Here is a brief replication cycle of the Newcastle disease virus, that how the F and HN proteins binds to the receptor, and in one way it leads to the formation of the endosome, and in the second way it inserts the nucleocapsid and input of the viral genomic RNA. Then negative stranded is converted into the positive stranded in one way, leading then negative stranded RNA leads to the replication, and then it is converted into the positive stranded and leads to the formation of the different proteins. And these proteins are processed by the Golgi bodies and the Golgi endoplasmic reticulum, leading to the formation of the new viron or the new virus. Newcastle disease virus cause damage to the different tissues, and those includes the pancreas, digestive system, or you can say gastrointestinal tract. Then it also causes the respiratory symptoms, signs, and also cause the damage to the respiratory system. Here, this picture shows that Newcastle disease virus at 1, 3, and 5 dp leads to the apoptosis into the pancreas. This slide shows that the how Newcastle disease virus in the vaccinated flocks. First column is about the control. Second is the vaccinated but with very low antibody titer, and those antibody titers were monitored up to 2 months after the vaccination. Then the birds were challenged with the virulent Newcastle disease virus, and we came to know that Newcastle disease virus caused the intestinal damage in almost all parts of the intestine, that is duodenum, jejunum, and ileum. But the supplementation of the vitamin E in the column C, in the column third column and the fourth column, decrease the damage caused by the Newcastle disease virus into the intestines. It also leads to the decrease in expression of the different amino acid transporters, then carbohydrate transporters. Then it also decreased the expression of the different mineral transporters. This slide shows that histopathological changes in the testes induced by the Newcastle disease virus, and these include the decrease in number of the Sertoli cells, and it can be seen from the slide B, and then individualized shrunken spermatogonia in the slide B and D. And in the slide D, we can see the multifocal segmented and spermatogenic columns, then necrotic cells, then lipid vacuoles. Lipid vacuoles are very clear. Even a person who is not histopathologist can see the lipid vacuoles and proteinous homogeneous material. Then, if we look at the current scenario, some countries are claiming that they don't have the Newcastle disease in their different regions, in their provinces, and those are Canada and the United States and the Western European countries. And when we look at the Asia, Africa, and the South America, outbreaks of the Newcastle disease are very common. Even the countries which are claiming that Newcastle disease is not present in their countries right now, it is common that the migratory birds that may be healthy, they can transfer the disease in those countries. So they are adopting different strategies to control it. For example, when we look at the United States, they have United States Department of Agriculture, and they have very good team that is working on the Newcastle disease virus under the leadership of Professor Afonso. And even they don't have, but they are working. And when we look at the world, this data or this figure is from 2005 to 2019. The red color represents the presence of the Newcastle disease virus, whereas the green color represents the absence of the Newcastle disease in those specific countries or specific regions. And just like black color, the upper one indicates no information. And when we look at the Pakistan, you can see in the India it is present, in the China it is present, in the Pakistan, especially in the Potoha region or in the upper region, in the hilly areas, it is present. I know many cases of the Newcastle disease in the central Punjab, in the Sindh, in the southern Punjab, but I think these are not reported to the OIE. And here is a table that is indicating the presence of the Newcastle disease in the American subcontinent and the different countries, including Argentina and the other countries of the America. And similarly, in these slides indicates the presence of the Newcastle disease, especially in Colombia. The presence is very high, or you can say it is present almost from the 2015 to 2005 to 2019. But when we look at the Greenland or the French Guiana, that it indicates that it has never been reported in these continents. But when we look at the Africa, the disease is very common and it causes huge economic losses. This may be one of the reasons that the United States Department of Agriculture funded give the funding to the UC Davis to do some genetics related research and find some disease resistant birds and then breed them for the African countries. And here, again, you can see the Newcastle disease is present in almost all the countries, especially in the domestic birds. These slides are about the presence of current scenario of the Newcastle disease. Newcastle disease have not been reported from the 2009 to 19 to onward. The reason may be the pandemic of the COVID-19. Then when we look at the Asia, Afghanistan, first of all, our concern is about the Iran, Afghanistan, China, and India, because these are our neighboring countries, and we have to look at the Newcastle disease or the burden of the Newcastle disease virus in those specific countries. And when we look at the Afghanistan, from 2000 to 2019, the disease was present in the Afghanistan. Then in Bangladesh, the presence is very high. And when we look at the other countries, Cambodia, not present. It was present from 2018 to 19. But when we look at the China, People's Republic of China, disease is present, but it is present into the limited zone and not in all the regions, or you can say it is not present in all the provinces of the China. Here you can see from 2019 to 2021, disease was not reported. I will discuss a few of the papers, and I know practical situation of the China. It is very difficult to isolate the virulent Newcastle disease virus from the domesticated, or you can say from the commercial poultry. And the reason is the vaccines, and we will discuss in the coming slides that how the Chinese have controlled the Newcastle disease in their commercial farms or from their commercial poultry, how they are preventing it. And when we look at the India, Indians have not provided the information to the OIE from the 2019 to 2021, but it is present into the Indonesia from where it had been started in 1926. And when we look at the Iran, that is very interesting. It is present from 2017 to 2019, but after 2019, information is not present. So these were our major concerns concerns in our neighboring countries. And when we look at Pakistan from 2007 to 2020, disease was present in the limited zones. I think it is present, according to my experience. I think it is present in all the regions of the Pakistan. Maybe it was not reported by the field veterinarians to the international organizations. That's why data is not present. Then we have to look at the immune system. I think all of the students or the professionals of the life sciences or the veterinary sciences or animal sciences, they know that naturally we have the innate immune system that recognize, that respond and create the antiviral state within the cells. If it do like this, then how NDV evade the innate immune responses? Naturally, NDV have evolve different strategies to bypass the innate immune responses. We will discuss in detail. Then NDV have the, in case of the vaccination in the commercial poultry, antibodies are produced. And mostly in Pakistan, we are using the live vaccines derived from the genotype 2 class 2 of NDV. And I think, according to my experience, that class 2 class 2 genotype 2 live vaccines of the NDV are not enough to control the virus shedding in the commercial poultry. That may be one of the reasons that NDV evades the adaptive immune responses, but we will discuss in the coming slides. Then we have to look how we can control it. First of all, NDV and innate immune responses. Innate immune responses, most of the people or the participants here knows that one of the two main immune strategies found in the vertebrates are innate and adaptive. This is the innate immune system provides the innate defense against all the pathogens that may be bacteria or the viruses, and its main function includes removal of identification and removal of the foreign substances, then activation of the adaptive immune system, the recruitment of the immune cells to the site of infection, then acting as a physical and chemical barrier. In case of the bacteria and viruses, toll-like receptor, then LNLR pathways are activated, and similarly, in case of fungus and RNA or the DNA, after the sensing of the RNA or DNA, different pathways are activated, and these pathways are described in the slides. But we will go into the specific, we will come to our specific topic, that is the RNA virus, negative standard RNA virus or the RNA viruses. In case of RNA viruses, viruses are sensed by the RIG-I or the MDA5. The most important thing in case of commercial poultry of the Pakistan or in the world, the chickens don't have the RIG1, RIG-I. Chickens only have the MDA5 and toll-like receptor 3 for the recognition of the RNA viruses. Only ducks, although ducks or the waterfowl is not present in Pakistani, in the commercial Pakistani poultry industry, but that is the biggest industry in China. But that may be one of the reasons that the presence of the RIG-I and the adaptation of the NDV virus by the activation of the NDV virus to the production of the interferons by the different way may be one of the leading cause of the decreased mortality, or you can say decreased outbreaks of the NDV in the waterfowl. After the sensing MAS, that is mitochondrial associated antiviral protein signaling pathway, signaling protein that is activated, that acts on the DDX3 and then TBK1 and then interferon regulatory factor 3, then production of the interferon. That is the common pathway, and NDV exploits this natural pathway that is present into the cells of the chickens by different ways. Then goose have the RIG-I pathway, and this RIG-I pathway, we have found this, we have sequenced, found and compared the RIG-I pathway of the goose, or you can say waterfowl, with the available sequences of the human or the mammalians. And we came to know after infection, sorry. Goose rigae and MDF5 are virus-inducible genes, and we have confirmed by the infection of the HeLa cells with the HERS33, ZJ1, and Lesota, and came to know that both the HERS33, that is the classical virus that was being used in almost all of the world for the pathogenic experiments of the Newcastle disease. And then ZJ1 is a wild type virus, wild type genotype 7 virus. And so we have infected the cells with Lesota, with ZJ1 and HERS33, and came to know that both the pathogenic viruses leads to the increase in expression of the rigae messenger RNA. Then at the same time, the expression was very less in the Lesota group. Then NDV also upregulated the rigae messenger RNA in vivo into the respiratory system. And it is also, you can see the infection caused by the HERS33 or rigae and SPF chickens leads to the increase in expression of the rigae in the lungs and also in the air sacs in a time-dependent manner. We have other evidence, and then this paper was published by our group in a leading journal of virology. And we came to know that ASAC is one of the biggest reason for the mutations of the Newcastle disease virus. For the natural mutations or recombinations of the Newcastle disease virus, and we
Idil take da garawan de meeting ne bhai. Please मुको बस
and it has decreased the mean death time of the specific pathogen-free chicken embryos, and it leads to the increase in pathogenicity. And so we came to know that sex may play an essential role in the antiviral immunity, because passaging into the other organs did not have significant effect on the viral mutation or the formation of the pathogenic viruses from the avirulent viruses. And we also described that the goose mouse mitochondrial associated antiviral protein signaling activates the interferon beta in species specific manner. And then how it works in chickens virus, because in chickens don't have the regi, and how it is activated in case of mouse is activated in case of chicken. So we came to know that NTV and AIV infections to the cells lead to the expression of the sting, chicken sting, that indirectly increase the expression of the mouse. Then we have performed the different cellular experiments, and we came to know that chicken sting inhibits the virus infections. And this was the working model. Then we have described some other pathways that how Newcastle disease virus cooperatively disturb the fission homeostasis to enhance the mitochondrial function by activating the unfolded protein response of the endoplasmic reticulum, leads to the increase in expression. And from these studies, we came to know that the main damage caused by the Newcastle disease may be from the oxidative stress. And we also studied the V protein of the Newcastle disease virus and came to know that the V protein is mainly responsible for the degradation of the mitochondrial antiviral signaling protein, and then it may lead to the inhibition of the host type 1 interferon production. So this is one of the viral pathway to escape from the innate immune responses of the cell. Then we have described these phenomena in these two articles, and one is because we came to know the main pathway may be the oxidative stress or the antioxidant system of the cell. So we have infected the chickens with the Newcastle disease virus and supplemented them with the vitamin E, and we came to know that it leads to the decrease in the tissue damage of the intestine. And then we have explored again about the virus, this pathway, and we came to know that Newcastle disease virus cause the double standard breaks, DNA double standard break response that facilitate the oncolytic Newcastle disease virus that is being used in many laboratories to treat the tumor cells. And this is one of the biggest benefit of the Newcastle disease virus, that it can replicate in the human cells, but only in the cells that are deficient in the basic innate immune responses, just like the production of the interferons. So we have proposed this mechanism that was published in Oxidative Medicine and Cellular Longevity, and this was the main pathway that was conclusion of 4 or 5 articles in the same row or on the same topic. And this slide, you can see from this slide that the infection of the hillia cells or the different cells by the Newcastle disease virus, just like Newcastle disease virus leads to the degradation of the MAFs. That is the main antiviral protein present within the cells that leads, or you can say that supports, that give the signal for the production of the interferons, especially type 1. In the first, and these are the results from the Western blotting, and you can see in the A, cells are mock infected, NDV infected, and we collected the samples at the 6-hour post-infection, 12 hours, 18 hours, and the 24 hours. And we came to know after infection, we can detect the NP protein of the Newcastle disease virus after 18 hours, and but it cannot be detected in the mock infected cells. But when we look at the mouse, and you can see in the subsection C of this figure that mouse is present in mock infected NDV infected up to 12 hours. But at the 18th hour post-infection, mouse cannot be detected from the NDV infected cells, indicating that mouse have been degraded by the Newcastle disease virus in the cells. I will not go into the detail of this. Then the other mechanism is that how that Newcastle disease virus leads to the cleavage of the DDX21, that is an other protein that is mainly involved in the antiviral innate immunity. And we have described, and you can see in the section B of this slide, that NDV infection, after NDV infection, we have collected the samples of NDV infected cells and came to know that at 24 hours post-infection of NDV, you can see in the DDX21, it is degraded, and its degradation leads to the decrease in size, or you can say change, or you can say it is converted into the two proteins. And this cleavage impairs the function of DDX21, leading to the escape of the Newcastle disease virus from the cells. And this was the proposed model. And then another question, this was some basic information that how NDV bypass the basic innate immune pathways to enhance its replication. Then the second major question may be that how NDV differs in their pathogenicity. Here, in the next slides, I can give you an example, because this example is from the genotype 2 Newcastle disease virus, and both are from the same genotype, but they have the different pathogenicities in ducks and chickens. And the results indicates that almost all the parameters, and that may be the hemagglutination, that 50% tissue culture infectious dose, then embryo lethal dose, or you can then the mean death time and intracerebral pathogenicity index in the old chicks. Almost they are comparable. But there is a lot of difference between their pathogenicities, and then why there is a difference in their pathogenicities, we have performed the sequence analysis, and from this slide you can see the replication of the NDV in the duck embryo fibroblast cells and the chicken embryo fibroblast cells. And as the name is indicated, the viruses that was isolated from the chickens replicates well in the chicken embryo fibroblast cells, whereas the virus which was isolated from the duck replicates well in the duck embryo fibroblast cells. But when we have challenged the ducks and the chickens with these viruses, we came to know that the chicken origin virus causes 70% mortality in ducks, whereas the duck origin virus, that with both the viruses belong to the same genotype, but the duck origin virus causes 20% mortality in ducks, whereas the ZJ1, that is a wild type genotype 7 virus, causes 10% mortality in ducks. These results indicate that Newcastle disease virus differs in their pathogenicity to the chickens and the ducks. And then we have described the histopathological changes induced by the different isolates in the intestine, trachea, lungs, and spleens. And here the phylogenetic analysis to show you that both the viruses are very close, and both belongs to the genotype 7. Then where is the difference? We performed the sequence analysis and came to know that there may be some point mutations at the 16 in the HF protein and HN protein, The point mutations in the F proteins are shown in this slide, and these are indicating that the duck origin virus have the mutation at 129, 179, and 181 and 396 residues. And similarly, chicken origin virus also have mutations. And then we perform the crystal structure of the NDVH protein, because this is the main protein that is involved in the receptor binding of the Newcastle disease virus, inside for the replication of the Newcastle disease. And we came to know that some mutations are very important, because those are present on the receptor binding sites, especially the mutation at the residue of 514. And the conversion of the valine to the isoleucine amino acids that may have increased the receptor binding, and then it may have increased the pathogenicity of the chicken origin virus into the ducks. Then adaptive immune responses, when almost all the participants, I think, knows that vaccines produce the antibodies, and how the different cells cellular immune responses participates in the elimination or in the removal of the foreign antigens from the different cells. But adaptive immune responses, as far as antibody titers are concerned, it varies according to the age, type, genetics, vaccine, health status, and biosecurity of the birds. Here in this slide, you can see the recruitment of the intestinal goblet cells into the intestine after NDV infection. And this recruitment of the different cells shifts the shift the different proteins that may be used for the increase in growth or increase in egg production, or for the that may be being used for the egg production or the increased growth rate. Those are shifted. Those energies are, in simple words, those proteins, those amino acids, and those energies are shifted for the recruitment, for the increase in cells in the intestine. One is the goblet cells. Second is the recruitment. In this slide, you can see the recruitment of intestinal enteroepithelial lymphocytes in the NDV infected cells in the P slide. And it's very clear. I think even the people who are not histopathologists, yet those can observe from the slides. And here, that is the important point, that recruitment of the mast cells. Although mast cells perform very important functions, and we have published a review on these cells, that is mast cells and innate immunity, master troops of the avian immune system. But recruitment of the mast cells into the intestine after NDV infection leads to the damage, and especially that damage leads to the decrease in expression of the different transporter genes, and can also lead to the leaky gut. That leaky gut may enhance the chances of the secondary bacterial infections in NDV infected chickens. Then our question is that we are vaccinating our chickens with genotype 2. Lesotho is very common in Pakistan, or Lesotho-derived live viruses are very common to protect the commercial poultry in Pakistani industry. But many research articles, or many, even the field veterinarians, if anyone is field veterinarian, and he may know, field outbreaks are very common even in the vaccinated flocks. And the similar results were published by the different scientists, that Lesotho vaccination may not protect against the lesions of the allergenic Newcastle disease in chickens. And similarly, characterization of the Newcastle disease virus from the vaccinated chicken flock in eastern China, it indicates that Newcastle disease virus, or you can say pathogenic Newcastle disease virus, replicates well even in the vaccinated flocks. Then, if it replicates well, what should be the control strategies? We should look at the production of the genetically resistant strains. No one will do this work for us. Even sometimes, I have a discussion with many field veterinarians or many industrialists, and they ask us for the final solution. Solution cannot be provided within a month, within 1 or 2 months. If we can provide, then it may be very time limiting. It may be for 1 or 2 months. But for the long-lasting solution of the Newcastle disease in Pakistan industry, we have to look at the molecular epidemiology of the Newcastle disease. We should know that should know the burden, type, genotype of the Newcastle disease virus that is causing the outbreaks in the different regions, even in the different districts. Then we can design, we can develop the good vaccination program or the strategies. For the vaccination, commonly used live vaccines, then the killed vaccines, and then genotype matched vaccines. This slide shows that even in the part A, you can see that is natural virus, and there may be some point mutations at the epitope sites of the, in the B virus, you can see there may be only three sites. There may be some mutations, but those sites are present on the cellular proteins which are mainly involved in the binding. We know that if some mutations are present, then there may be more utilization of the antibodies, or you can say antibody titers. That may be one of the reasons that the antibody titers of our commercial flocks may be more than 10, more than 12. But even with that titers, we experience the replication or the decrease in egg production or nervous signs, or even sometimes mortality in commercial flocks. So we have to look at the genotype matched vaccines. One way is the formation of the recombinant vaccines using the backbone of the Lesotho or using the backbone of the genotype 2 viruses. And second way is that you can develop the killed vaccine. That is the most economically easy and less technical way. Here you can see the comparison of the virus shedding with the different vaccines that may be genotype matched and not genotype matched, which were not genotype matched. And there is a clear-cut difference between the different groups at the 5 and 7 DPI in chicken and geese, and in the genotype matched and genotype matched vaccines. You can say genotype matched vaccines decrease the virus shedding from the trachea and from the cloacal swabs. Then comparison of the virus shedding by the different vaccine, that is oil, simply not vaccinated, Lesotho, and then recombinant growth, and the mean titers of the swabs immunized on the sampling days 0 to 7. Then here you can see the frequency of the isolation of the virus challenge. Here are the different groups. One and this experiment was performed by the Dr. Milner. She is very well known among the field veterinarians or the persons or the scientists working on the Newcastle disease. She is one of the leading lady in the world on her work, due to her work on the Newcastle disease. She have developed the different vaccines from the different viruses. And then she have challenged different groups with the different viruses that may be of different origin. And according to her results, you can see virus shedding in the Lesotho after Malaysian challenge was higher, and it was decreased when we are going to use the genotype matched vaccine. For example, when we have challenged the virus with a Malaysian strain, and also developed the vaccines of the vaccinated the chicken with the same killed vaccine. We know that from the results it is indicated that these birds or this group have the minimum virus shedding. There are different views of the scientists about the control or about the use of the genotype matched vaccine. One group is saying that we should focus on immunity, we should improve the immune status of the birds, and then we can use the Lesotho vaccine. Some groups are some scientific groups or the researchers are saying that we should use the genotype matched vaccine. And according to my experience, or according to the experience of our lab, that it is the best way to use the genotype matched vaccine to control the Newcastle disease. Only few slides are remaining. Then, what will be the effect of genotype match vaccine on the virus evolution? If someone is sitting from the virus evolution or immunology, he may know that the vaccine-induced antibodies may force the viruses for the evolution. So question is that we are using the genotype 2 vaccines, and those vaccines are not causing the virus evolution. Practically, they are not causing. But if we will use the genotype 7 vaccine, or you can say genotype match vaccine, what will be the effect on the virus evolution? So to know this, we have performed some cellular experiments, and results are published in the journal Poultry Science. And the results indicate that even the use of genotype 7, or you can say genotype match vaccine in case of Newcastle disease virus doesn't lead to the too much increase in the mutations of the viruses. So these are the results and the results are showing non-synonymous to synonymous mutations in the F gene of the different viruses, that is GJ1 and ND120, without in the different group. That one group was without anti-serum, second group was with anti-2 serum. Anti-2 means in genotype 2 or Lesotho-based serum. Then the third group was anti-7 serum, that was genotype match serum. And we came to know that the mutation or the ratio of the non-synonymous to synonymous was not alarming if we are using the genotype match vaccine up to 30 generations of the virus. What will be the future threats? We should look at class 1 NDVs. When we look at the percentage of the avirulent viruses present into the different migratory birds, or you can say in the different birds, non-commercial birds, or even in the waterfowl, chances or the number of NDV, class 1 NDVs are more compared to the pathogenic NDVs. According to experience of our lab, the pathogenicity of class 1 NDVs is increasing. And that increase in pathogenicity, especially the mean death time into the specific pathogen-free embryonated chickens, is alarming, and that can cause the epidemic, or you can say pandemic, maybe after 10 years or 15 years. We have the record of last 5 years, and in the last 5 years, many of the class 1 NDVs have changed their nature, and their mean death time decreased from 130, 140 hours to 120, or you can say even in some cases 115 or 110. But if it is decreased up to 70 or 60, then it means that is a pathogenic virus. So how we can prevent our birds, how we can do the precautionary measures? We should decrease the chances of contact with the waterfowl, because that is the main harbor. And secondly, live bird markers that play an important role in the spread of the class 1 viruses. And I think in Pakistan, there is not any policy about the live bird market. That, according to my point of view, that is one of the biggest risk for the Pakistani poultry industry in future. And then the fourth one is the rural poultry. In rural poultry, we are not vaccinating our chickens or our domestic birds against the Newcastle disease virus. And in case of outbreaks, those birds can spread the virus to the commercial farms or the other communities. And at the end, in summary, Newcastle disease virus degrades MAVs, DDX1, and exploits the autophagy and induce the oxidative stress to bypass or to evade the innate immune system. Then NTV and adaptive immune system. NTV shift the energies or the proteins or the best quality proteins of the body from the production of eggs or increase in growth to the recruitment of cells to the and leads to the damage to the different tissues. And then genotype match vaccines can decrease the virus shedding. Then for the control strategies, we have to look at the genetic selection. We should find the genes and then those genes may be included in the breeding programs. And then we should develop the genotype match vaccines. And then biosecurity is very important. Then the health status, and then some herbs or the phytogenics have the promising role in the decrease in the replication of Newcastle disease. So that was the end. If you have any question related to the Newcastle disease or their interaction with the innate or adaptive immune responses or how we can control it, please let me know.
Thank you very much, Dr. Zaibur. And now we have the question answer. We can take 5 questions. If any person have any question, can raise his or her hand, or also can write in the chat room. Any participants, any student can ask for question.
I think it will be more interesting if you can ask some questions related to Newcastle disease.
I think there will be no question. Dr. Zaibur, thank you very much for sparing your time. It was a very wonderful talk on the novel diseases and latest issues. Hopefully this will be a great opportunity for the participants to learn about this disease. And thank you the participants to be with us for 1 hour. And sorry for the light issue. Today there was some light fluctuation. I am again thank you the speaker Dr. Zaibur for sparing his valuable time for this presentation. And our next talk will be on the 28th October, and the topic will be about the variable rate spring system. It is about the precision agriculture. I will request the participants who have interest in the precision agriculture can also participate in that talk. Today our seminar will be ended, and again thankful to the speaker for his time. Thank you, Dr. Zaid.
Thank you, sir. Thank you, sir.